cytokinins discovery. Here cyto means cell, kinins division, kinesis is division. So cell division promoting substances, cytokinins. First, a Skoog scientists observed the tobacco plant internodes. From this here it is internode, completely internode. From this internode, some tissue is taken. So this tissue is cultured. So when the tissue is cultured in the tissue culture medium, so it is proliferated, or it is producing the new cells. When in the tissue culture medium, it is provided so it is tissue culture medium. So from the tobacco plant internode, here the tissue is taken and it is cultured in the medium. It is showing the cell division rapidly. So when the medium is provided auxins, in addition to auxins, cytokinins also. Here, in addition to auxins, some cytokinin containing substances like coconut milk, or here extract of vascular tissues, sometimes here yeast DNA also, yeast DNA extracts. So in the presence of auxins, so in these components anyone, so auxin and these component, any one component. So here auxins and one of these components, when they are provided in the medium, so the tissue, it is here showing the cell division fastly and it is developing into the new plant. So that was observed by Skoog scientists. Skoog, here he observed the tobacco, he extracted the some tissue from the tobacco internodal part and it is cultured. So this medium is provided with auxins. In addition to auxins, one of these components are also added. So the tissue is showing the cell division very fast and that is developing into the new plant. So if these components are absent, the tissue is not proliferated. So means here all these components are responsible for the cell division. The cell division promoting substances are present in these components. And later, Skoog and Miller scientists, extracted the cytokinin. The cytokinin are cell division promoting substance from here. Cytokinin are cell division promoting substance. It is extracted. It is named as kinetin. From yeast DNA, coconut milk, extract of vascular tissues are having the cell division promoting substances. So that's why that substance is named as kinetin. This kinetin was firstly isolated or separated by these scientists, Skoog and Miller scientists. And Miller and Latham scientists also extracted the one more cytokinin. Miller and Latham scientists. These scientists extracted the one cytokinin here, that is zeatin. Here the kinetin is extracted from herring, fish, sperm DNA, from DNA, and zeatin is extracted from Here GEAMCET, it is from animal, fish. Here the gelatin is from plant, GEAMCET or corn plant seeds. So Skoog scientists observed like this and Skoog and Miller scientists extracted the cytokinin, that first isolated cytokinin, it is kinetin, but the kinetin is not natural. Miller and Latham scientists extracted the one more cytokinin, that is gelatin. It is natural cytokinin. Natural cytokinin. It is not natural. Why? Because the kinetin is produced or released by the DNA when the DNA is destructed. Here the DNA, when the DNA is destructed, then only it is synthesized. But naturally the gelatin is present in the corn seeds. So it is about the discovery of cytokinins.